Hello, everybody. Welcome. This is the How To Talks by Postdocs Spring 2021 mini series. Today we have Dr. Liza Roger. She will be talking about using environmental engineering to benefit coral reefs. So Dr. Roger grew up on the coast of Normandy, where she loved playing in rock pools at low tide and became passionate about the ocean after seeing her first underwater documentary. Dr. Roger studied marine biology and coastal management in France and obtained her Ph.D. in Western Australia at the Institute of Marine Science. After graduating, she continued to explore all things ocean related, including being a whale watching tour guide in Iceland and a scuba instructor in Greece. She joined Dr. Lewinsky's lab last March and has enjoyed getting into her new projects. So today we're going to hear about one of those, and I'll let her take it away.
Hi, thank you, Stacy. So yes, I'm going to talk about how we're trying to help coral reefs using nanotechnology and nanoparticles especially. And because of you might be familiar with how the how-to talks work. I'd like to give you a heads up. It's not a typical how-to talk. I don't expect you to go home, make nanoparticles and go and save coral reefs. But it gives you an idea of how we are trying to do it. So first, I will give you some background on coral reefs, what they are, what they do, and the threats they are facing. And then I will jump into what we're trying to do. So here we go. So first of all, what is a coral reef? Well, as you can see on the images here, they can look very different depending on the location. The coral reefs span quite a lot of distance, and they're concentrated between the tropical and subtropical regions around the globe. So quite shallow waters, quite warm, and there's a lot of light. This is conditions that they need. And as you can see here, you've got some very different shapes and colors, different organisms that are involved, different types of fish. And you can see the different locations from Florida, Japan, Maldives, Fiji, American Samoa, Australia, India, Indonesia, Mexico, and that's not an exhaustive list. These were just really nice photos that I wanted to show you to give you an idea of how pretty it can look. If you ever have a chance to go and dive or snorkel on a coral reef, definitely recommend it, because it will change your perception of the sea. Most people see the sea as something very dark, but coral reefs are far, far from that. And so we can call coral reefs cities of the sea because there's so much life there. And like cities, you can actually see them from space. For example, this is a satellite image of the northern part of the Great Barrier Reef. And yes, you can, the sort of turquoise structure that you can see along the edge there, this is the Great Barrier Reef. So like human-made structures, you can't see them from space. They also have this great ability to provide ecosystem services. And by that, I mean they provide food, shelter and protection for other organisms that live there. They provide a community, genetic diversity, and they are self-sufficient. So this is very important. And so this is all in terms of the organisms that live there, but they also have great value for us as humans. So we get a lot of our compounds that are used in medical research and treatments from marine organisms. The reefs provide protection along the shore because they can dampen the energy of the waves and sort of pick up the slack in terms of flooding and when there's large weather events. They also provide a lot of income from tourism and food as well, so from the fisheries. And of course, you know, on a more sort of planet scale, they have a huge biodiversity. It's 25%, which is pretty big. Now, coral, so I was talking about the structure up to now, but if you look at the more specifically, what is at the center of a coral reef, it's coral. So you have different types of corals that form different shapes. So this is one of the main classifications we use to know what's what, basically. So you have the branching corals, you have the mount forming corals, encrusting corals, plate corals. it's amazing to see the diversity of things and how they can grow, because they totally do that on their own. And if we look at, so a coral colony here, so this would be a branching coral colony. You zoom in, you see a little coral polyp. So the polyp here with the tentacles on the edge, and that's quite small. That would be maybe 1 millimeter. Then if we look at the anatomy of the polyp itself, so you can see here, like I said, you have the tentacles, you have a mouth, an oral cavity, and then you have a whole bunch of other polyps along the surface, because they don't live alone, and they form colonies, and those colonies are made of their own clones. So they're all the same on the colony. Now, the way they build these reef structures is by forming calcium carbonate skeletons. So calcium carbonate is limestone, basically, or chalk, whatever you want to call it. And so the coral, as it grows, it builds layer by layer by layer of this skeleton, of the structure underneath. And the tissue itself, if you compare, so if you imagine you're wearing a latex glove, the thickness of the glove would be the amount of tissue that is on top of the skeleton. Now, the really cool thing that they also have, these corals, is they have symbiotic algae that lives in their tissue. The symbiotic algae are plants, so they perform photosynthesis, and they share the product of the photosynthesis with the coral. That also enhances the growth of the coral, so you get bigger structures. So that's why they also need light. And then an extra layer of complications is the coral microbiome. Like humans and other animals, we have a microbiome, so we have the bacteria and viruses and microbes that live within us that don't make us sick most of the time, but we all live together in a nice, happy community. Well, it's the same with the corals. Most of the time, it doesn't make them sick, but they have a very diverse microbiome. And then, because, as I mentioned before, we're trying to help them out, there's clearly some threats on coral reefs, and here are a few of them. So you'd have fishing practices, pollution, of course, human activities mostly related to leisure. Then you'd have industries, oil and gas are pretty bad, coastal developments, because everybody wants to live by the sea. And then, of course, the big winner, climate change. So the way we're trying to help the coral reefs is to try and mitigate the effect of climate change at this stage. And for example, ocean warming is the main issue at the moment. It's the most pressing problem, actually. So here on this map, you can see the heat content of the ocean between 1993 and 2019. So the orange, reddy colors are where there's been increased heat content, and the blue where there's been decreased heat content. You can see, for example, around where we are on the coast, it's quite red orangey, so a fair bit of heating, but then around the tropics, so towards the Caribbeans, it is sort of pale orange. So you wouldn't expect a huge increase, but don't be fooled. This is quite significant for corals. They can't really handle this type of heat. And then here's another version of this heating, more on a graph, and gives you a time scale, again, over the same time period. And the red orangey colors are the surface waters, so the heating of the surface waters over this period, and the blue are the deeper waters. So less heating in the deeper waters, but much higher heating in the surface waters. And the corals are usually in shallower waters, especially if they want to do photosynthesis. And so the main issue with ocean warming is the fact that it triggers coral bleaching. When a coral is nice and happy, it is colorful. So, and it doesn't mean it's bright orange. Some of them can be bright purple, or bright blue, or bright pink, but it's not, it's quite a normal color for them to be sort of brownish. And then when there is stress, and here we are more talking about thermal stress, the algae that lives in their tissue leaves the tissue. And that's how the coral becomes white, because they still have tissue, but you can now see the skeleton underneath the tissue through the transparency of the tissue. And this is what we call bleaching, and this is a big problem because they can't sustain themselves like that. They need the symbiotic algae to live. They can still feed themselves because they have other way to eat, but it's not enough for them for their needs, basically. And to give you a sort of visual of this, so this is a photo that was taken in American Samoa in December 2014, and a healthy reef. You can see, like I said, it's not a bright color, but this is a healthy reef. So slightly brown orangey, got some little colored tips, so slightly blue purple. And a few months later, the coral bleached because there was a heat wave. So this is in February 2015. And again, a few months later, everything is dead, August 2015. And so you could think that this color here in August is quite similar to the first one, so maybe they're still alive. But this is actually macroalgae, so a different type of algae, more of the algae that grow on rocks that now has started growing on the skeleton after all the tissue came off. So the coral has died and something else has come and moved in instead of it. And it's not a new phenomenon, coral bleaching. It's happened before, and we have recorded previous events of this, but it's happening faster and faster because warming of the ocean is happening more and more. Now, to get more into detail in the bleaching mechanism, so you'd have your happy coral, a nice brown color, then you apply the heating. So what happens is they go through what we call oxidative stress. They have too much reactive oxygen species. These are molecules that react very fast and are based around oxygen, most of them, and normally they have a coping mechanism for this. They have other enzymes and other molecules that come and control this oxidative stress. The issue that happens with bleaching is that the oxidative stress is too high compared to their coping mechanism, so they can't handle it. And what happens after that is they lose the symbiotic algae, they're all bleached. Case one, like I showed you before, the temperature is still high, they can't handle it over time, they die, and macroalgae comes in and takes their place. The other option is the temperature drops again. They are able to get more algae in their tissue, so more symbiotic algae, or they had some left, and because the temperature dropped again, the microalgae finally managed to multiply again and share its benefit from the photosynthesis with the coral again. So, case two is much better clearly than case one. They're still not as healthy as they were in the first place, but they are recovering. So the steps where we can potentially intervene is at the oxidative stress stage, or to prevent total death and trigger recovery. So that's what we're trying to do. Now, if we look at oxidative stress, the different ways that we can treat this, for starters, oxidative stress, we have it also as humans. This is what can trigger a fever, for example. So if we compare to the human system, you have a happy little human, oh, it has a problem, it has a fever. So not so happy anymore. But then what do you do if you're ill? You take medication and hey, you feel better. So if we compare this to the corals, we have the corals, it's got a temperature, it's got a fever. So it bleaches, unhappy. And then we give it a treatment and the coral recovers. So that's what we're trying to do. We're trying to treat them. And the way we're trying to give them this medication is by using nanoparticles. Now, nanoparticles. You might not be aware of what it is, and I'm quite new to this myself. But basically, they are very small particles. And this is the nanometer scale, for example. And I'm going to plot for you a few different molecules on this scale, on molecules that you might know or might not know, but to give you a size range of what we're working with in terms of the nanometer scale. So you have water, glucose, antibodies, viruses, bacteria, cancer cells, a full stop. You know, when you're typing, like on a computer, a stop, that's the, or what do you call it? Period, right? In a sort of American, a period. And then you have a tennis ball. So on the scale we're working with for the nanoparticle is here, is 1 to 100 nanometers. So from below water size to up to virus scale. And then the good thing with the nanoparticles is that they have a huge surface to weight ratio. I mean, good and not good, depends. And they have very specific properties that can be different from the original material. And then they've been applied to things like putting a coating on your glasses so they don't get scratched. They have been made fluorescent for medical imaging. They've been used in anti-scratch paint or anti-fouling paint. They've been used in clothing. So there's many, many applications for nanoparticles. Biofuels as well, so many out there. So we are looking at antioxidant nanoparticles. And because the oxidative stress is what we are trying to fix. So if the antioxidant, we are hoping to fix the oxidative stress that way. So what do we need the nanoparticles to do? We need them to enter the organism. So through digestion or through the skin, sort of the skin. Then we need them to enter the cells. So there's different ways they can enter the cells. So through specific receptors, pinocytosis or phagocytosis. We need them to act as antioxidant. And of course, we need them not to kill the coral. That's best. And so now I'm going to go through each of these to try and explain how we're trying to get to that specific objective. So entering the organism. So that's the delivery method. How are we going to get the coral to take up these nanoparticles? So the first one would be digestion. So you have a little polyp, and a good idea would be, let's just use a syringe and inject the nanoparticles. Except if you remember before, I said that some polyps can be very small. So 1 millimeter is like this species on this photo. You have bigger ones, but if you're working with small ones, imagine having to treat every single polyp on one coral and then multiplying that by the number of corals that you have on your reef and the numbers of reefs that you have to treat. So on the ground, that's not really realistic, unfortunately. So another method would be mixing the nanoparticles with the seawater. And the corals have these little tentacles that they use to catch food, sort of water filtration. So they could very well just catch nanoparticles and eat them. That's an option. Now the other option is through the skin or the epithelial layer of the coral. So like this. But now, so you could, again, mix them in the seawater. And if they don't get caught by the tentacles, then they might just deposit on top of the tissue and go through. And the other option would be to spread a sort of ointment or a cream onto the tissue that would then help the coral absorb these nanoparticles. But then we're going already onto the tissue. And how does that work to get the nanoparticles to go through the tissue? So the first one would be between the cells, intracellular. And so you have your seawater at the top here where you have your nanoparticles. And here you have your coral tissue with here I just put 2 layers, so 2 layers of tissue and your cells and your nanoparticles. So intercellular, you have your little nanoparticles that go between the cells. Now, the other option is intracellular, and this time you have your tissue again, your nanoparticles, and they go through the cells. So this feeds into the other aim that we had, which was how do we get the nanoparticles to enter the cells, the intracellular transport. This is tricky. There are 3 different ways, more or less controlled. The first one is phagocytosis, where you have your cell, you have your nanoparticle that gets not ingested. The cells don't have mouths, but they take in the nanoparticle and create vesicles. Then you have pinocytosis, where it's very similar to phagocytosis, except they don't just take the nanoparticle, they take other things with it that are in the media. And the media, I'm talking if it's directly from the seawater, so it would be from the seawater. They take other things with it. So it's like seawater and particles. And then they have a vesicle again with all of this inside. And then the last one is receptor mediated. It's where you have a cell that recognizes your nanoparticle and specifically takes it up. because it wants it, it recognizes it. So this is quite a good way to target a delivery. And now how would you promote this delivery? So you would have, we specifically work with cerium oxide at the moment, because we already know that it has antioxidant properties and reactive oxygen species, that's the ROS, reactive oxygen species, scavenging properties. So it picks it up quite fast. And so the way we would promote the internalization of cerium oxide is we would add different coatings to the particle, not give it to the corals bare, but put a coating around it. So you give it different coatings to see which one is going to be preferentially recognized by the cells that you want the nanoparticles to be taken up by. And you can see that the targeted delivery means that the ones that don't have the right coating are not going to go in, the other ones will, and they will stay there. So that's quite a good way to do it. Now, once we have our coating, and I've explained to you we're using them and everything, but how do we make them? So again, I'm taking the example of cerium oxide, and there's many different methods of doing this, and I like more simple methods because it just makes life more easy. And so this is a simple way of making nanoparticles. So you would have your solvent, and then you add some cerium seeding crystals. This is what will start the nanoparticle nucleation. It's going to be at the core. And then you have your coating polymer. It mixes for 24 hours. You purify it, you filter it, and ta-da, you have your coated nanoparticles. I make it sound super simple, and in process it kind of is, but it's not an exact science. So you really need to characterize your nanoparticles afterwards. There's a few different instruments that can help you do this. So you have the DLS, dynamic light scattering, you have the TEM, transmitted electron microscopy, and XRD, X-ray diffraction. So these different instruments can help you figure out what size your nanoparticle is, what it's made of. So do you have the right type of nanoparticles, and also the shape. And you can also figure out, well, not figure out, you can also measure whether your nanoparticles will stay in suspension in the solution. So if you're using seawater to deliver the nanoparticles, you need the nanoparticles to stay in suspension in the seawater, because if they sink immediately to the bottom, then the corals are going to pick up very little of it, and they might potentially be smothered by the amount of deposition of the nanoparticles. So you want to avoid that. You want them to be in the water for long enough for the corals to or pick it up with their tentacles, or whatever way you've decided to deliver them. But you need them to be there. And then if you remember correctly, the next step was trying to make the nanoparticles so they don't kill the organism. And the first way, there's two steps to this. The first one is in vitro testing. In vitro means in cell cultures, basically. And the problem with corals is that we don't have immortal cell lines for them. We can't just order them online, get them delivered, they stay in the freezer, and we need them, we spring them back to life again. It doesn't work with the corals. We have to, every time, at the moment still, have a coral fragment, and then harvest a piece, get the tissue off, and then get our cells from there. So it's quite an easy step to do, but it does add extra work, and it adds if you don't have a good cell culture, then it's going to affect your results. And so what do we do with the cells to test whether the nanoparticles are going to kill them or not? This is when we do a dose response test. So you would have your well plate, which actually looks like this, the little images up top. And then you have your stressor, which would be the temperature here, the different doses of nanoparticles, and you want replication so that you can statistically confirm your results. So imagine that I have cell cultures of my coral cells in this plate here, and then I populate it with my different doses. And in these cell cultures, I also have a fluorescent dye that tells me whether there is oxidative stress or not. So let's say it starts fluorescing. If the stress is there, If it isn't, you don't have fluorescence, or you have very low fluorescence. And then, as you might have noticed, some of the colors here aren't on the dose system. So here I have one control, which would be the blue one, which would be the 0 micromolar, so no nanoparticles. But you need also other controls. So you need some nanoparticles without cells, and you need cells and nanoparticles without the dye. And this will help you to correct for background and potential mistakes, or to check whether there's an issue with or your cells or your nanoparticles or your media. And then you need a little bit of water on the edges to prevent evaporation. And it's better if it's the same osmolarity as your cell cultures. So for me here, if I was working with coral cells, I would probably put seawater. But here I put water just to give you an idea. Then afterwards, from there, you get this type of data. So you would get the fluorescence from the specific dye, and then you want to know whether your cells are alive or dead. So if you look at the fluorescence graph, you have the different concentrations. So the 0, which is the control, then all the other ones. Then you have the 2 other controls I mentioned before. So your different concentrations of nanoparticles are nicely sitting between your controls. And by the way, this is simulated data, so don't get excited. I haven't solved the problem yet. And then you can see that this one, so the yellow line, is the lowest concentration, apart from the 0, of course. Lowest concentration, and it is doing what you would want it to do in a perfect world. So you have an increase of the oxidative stress, so it starts fluorescing and then it drops again. So you could say that your nanoparticles are addressing the stress and making it drop again. And then if you look at this concentration on your viability graph, you can see that your cells are about 95% alive still. So that's a very good number. Again, simulated data in a perfect world. Yeah. So the next step after this, because I told you there was 2 steps. The first one was in vitro, and this one is in vivo. So you want to try the doses that worked in your cultures on the coral itself, the colony, because they might not work the same. You don't know. There's many more interactions than just the cells, because there you've just separated all the cells and all the tissue and concentrated on that. But when they're together, there's mechanisms that you don't necessarily know that are going on that might affect the response to the nanoparticles. And so we do tank experiments. For example, here, so this is a large tank in which you put sub-tanks. Here there's 4, but for replication, you'd repeat these 4 3 times. And so in 2 of them, you would have normal seawater as your control. And then in the other ones, you would have your heated seawater. And in both scenarios, you would have some that are treated with the nanoparticles and some that aren't, because you really need to know the response to this. And you have at least 3 corals in each tank, again, for replication. You need a light and temperature sensor to make sure that your temperature is the one you set it at, and whether there's fluctuations throughout the day. And you, with the light, you can check that you have your normal light cycle. Then you can also measure the outflow, so what is coming out of your tanks, to see whether there's a lot of nanoparticles in the outflow, because it might be that maybe, you know, the cells were taking up the nanoparticles in your cultures, but they're not taking them up so easily in the tank system. So you never know. And then drawings are all well and good, but what does it look like in an actual lab? Looks something like this. So this is not our lab. We're a little bit less fancy, but this is in a nice example of you have a big water table that has a large tank basically, and then you have the sub tanks in here with their own water system because they have their own treatment in each of them. And now the last step of this whole process is trying to figure out whether we can release, so if the treatment works on the cells and the treatment works on the coral colonies, you know, we want this to be applicable to the reef scale. So is it safe to be released in the environment? So it's a sort of environmental impact assessment. And for that, you need to answer a whole bunch of questions. So from how long do the nanoparticles stay in the coral? Do they dissolve in the seawater? Because some nanoparticles change through time, and seawater is quite a complex media because it has a lot of ions and salts in there, so it can modify compositions of stuff quite easily. Then do they accumulate anywhere? Because if here you've answered how long they stay in the coral, here you need to answer how long they stay in the whole system. Because as I showed before, there's a lot of different organisms that live on coral reefs. And most of the time, the places where anything accumulates is towards the bottom. And there's a lot of life on the seabed. So is this going to affect whatever lives there? So this is a question that needs answering. And then are they going to be toxic to these other marine organisms that live or on the seabed, or even fish? We have fish that eat coral. If the fish that has eaten a piece of coral that has nanoparticles, is the fish going to get sick? So this is the kind of thing we need to look at. Also, yes, should we then, to avoid potential toxicity to the other organisms, should we pick the nanoparticles up once they've done their job? As I mentioned, nanoparticles are very small. It's not a question of picking them up. You have to figure out how to collect the nanoparticles again. Basically, all of this is to figure out whether the nanoparticle is safe to be released in the ocean, because otherwise we did all this work for nothing, really. And this is intense. That involves a lot of work, repeating experiments on different life forms, and or having a mesocosm experiment, so where you have you simulate a mini coral reef in tanks again, but where you have many different species and many different organisms living there to simulate the diversity of a coral reef. Because you have the fish, the gorgonians, the starfish, the corals, everything that lives there. So clearly, it takes time. It's quite a long process. It's not going to happen overnight, and we haven't solved even all the previous steps to this yet. We're working on it. So until we do that, I will leave you with this, and I will I hope you can be more conscious of how you can help coral reefs to protect them and preserve them through sustainable seafood choices, saving water, being careful if you go diving or snorkeling, and type of sunscreen that you use, what kind of waste you produce. So all of this, until we fix the problems for the coral reefs, please be careful. That's me. So I want to say thank you very much for listening. If you want to contact me, you have my email up here. I want to say thank you to VCU Libraries for organizing the mini-series, to Dr. Lewinsky for bringing me to VCU. Otherwise, I'd still be stuck in France right now. The NSF funding, of course, and the huge team that we have on this synthetic coral project. So you have all some of the team members here, sort of the core. Then here are a few sort of links to articles that you might be interested in reading, and some of the blogs that we have. I am more than happy to answer questions if you have any. I guess you can put them in the chat if you want, or whatever works. Let's see, can I open the chat? No, I need to stop sharing to open the chat. Sorry. No questions in the chat.
Okay, well, thank you, Dr. Roger, so much for this very interesting talk. I had a question. What, if you had to forecast, and based on the work that you've done so far, what do you think the most successful delivery method would be?
I think at this stage to really target, because it's the same, so you have certain corals that respond differently to thermal stress. So you probably wouldn't want to treat them all if they're not all needing the treatment.
Right.
So I think the the ointment, sort of cream version of the treatment would potentially work well, because you can target specific ones. Yeah, and then if we make it one that can dissolve in the waters, then you don't have potential implications for other organisms. It's just a question of figuring out how long the nanoparticles stay in the system overall, really.
Great, thank you. And now I see we've got some questions in the chat. I'll read them to you so you can think about them. Deirdre asks, how guilty should I feel using sunscreen?
Well, there's some sunscreens now that are made to be safe for the reefs, because there's been a fair bit of research on this. And Hawaii and the States are actually leading the path on that. So it's quite good. And you can find them when you're already on holiday. It's usually a little bit tricky to get them. So I'd recommend thinking about it in advance. You've got some very nice ones that work really well.
Excellent. And then, so is this a long-term solution for the health of the corals, or just for more immediate cases?
Well, it would be nice if it was a long-term solution, because we don't know how long this problem is going to keep going, because it doesn't look like it's slowing down at this stage. We can, as a community, take actions to slow the rate of heating of the oceans, but it's not going to happen tomorrow. So this is why we need to find solutions that we can apply now. And then, if they have to keep going, they will keep going. But because it's still early stages in the development of this sort of technology, we need to figure out whether, if the nanoparticles stay in the coral, are they year after year going to provide this antioxidant treatment, or is it just a one-off and then you can't, they're done kind of thing. So this is another thing that we need to figure out. But it is, hopefully it's a long-term solution, because this type of nanoparticle, the serum oxide, has this potential to shift quite fast and quite often between two different states, where it can pick up or not the reactive oxygen species. So as long as we make sure that it go back into this stage of being able to pick up the the reactive oxygen species when it need to, then we should be able to help them on a longer term than just a one-off treatment.
Awesome, thank you.
Oh, sorry.
Oh, go ahead.
Sorry, I'm not turning my camera on because I had a workout right before. I'm Christine Van Dyne. I'm in a lab in the microbiology department, and I work with Pseudomonas, which I'm only bringing it up because it secretes this kind of biofilm that is made of alginate, like a seaweed derivative. I was wondering, with like the ointment that you're talking about, first off, do corals have kind of biofilms? Would you need to would that disrupt the ability of the biofilms to function, just like their natural, commensal organisms that they have? And also, would you need to cover the entire coral, or is it something that if it comes in contact with some of the coral, that would be okay? That's question one. Sorry.
So the corals produce a mucus layer. Depending on the species, it can be thick or not, and when they're stressed, they tend to produce more of it.
Okay.
So yes, there is a mucus layer that can affect this, and then the composition varies from one coral to the other.
Okay.
And then what was the other part? Sorry.
Oh, do you need to? Do you need to basically cover every single surface of the coral? Yeah.
So I think the idea would be a more targeted approach because the healthy part, you could assume that it is healthy, so you don't have to treat it, and you can usually see the bleaching in specific areas, and sometimes it is the whole thing. So if you have to treat the whole thing, then it would be the entire part. But I guess it would depend on the type of coral.
Right. Okay. And then my final question, and I've actually been thinking about this. I've been wanting to make a transition back into marine bio. I miss it a lot. So with Pseudomonas, they produce this alginate, this kind of biofilm that's basically a component of seaweed. And I'm wondering if you could put, I worked in nanoparticles too, and they you can use matrices to maybe remove free radicals from a stint in the body if you had a biofilm growing there and you wanted to treat it with nanoparticles. I wonder if you could have some kind of medium, maybe alginate or something like that would stay on the coral long term and slowly, would absorb. I know that's a really weird idea. I was thinking about it for a while when I saw your talk. Yeah. So
it is one of it's one of the possibilities when you were thinking about using 3D bioprinting to generate an alginate hydrous sort of mixture that could hold the nanoparticles that you would apply to the corals when they need treating. It would be fantastic, if it's something that could respond to environmental conditions. For example, if the heating in the seawater is sustained and sort of higher than normal, then the alginate would respond, and I don't know, the nanoparticles would shift toward the tissue.
Yeah, and the idea would be it wouldn't be a big deal if a sea turtle, chomped a little bit of the matrix because it's already a component of seaweed. So that was, yeah. So, okay, thank you so much. I know there are kind of off the wall questions, but I appreciate your time.
It's cool.
Thank you.
If you want to make some of that seaweed alginate for us, we can try it on the corals, no problem.
I will give that a shot for sure. I will be in contact. Thank you.
Great. Thank you everyone so much for coming. Next week will be our final offering of the 2021 How-To Talks by Postdocs mini-series, and we'll have Dr. Sean Cobley talking about practical Bayesian statistics. And so if that's something that's interesting for you, please make sure that you register today and visit our website. We thank again Dr. Roger so much for sharing her information with us and look forward to seeing you all again soon. Thanks and have a great day.